Our next speaker is Andrea Jurisicova, and she's going to tell us about animal models of reproductive aging and oocyte aneuploidy.
Good morning, everybody. It's really my pleasure to be here today, and I would like to thank Dr. Zhang for giving me an opportunity to come and talk to you about some of the work that we have been doing in a lab for well over 15 years or so. And so what I'm really interested in is trying to understand molecular pathways that are affected by aging, as well as what kind of mutations and genes could possibly plague human infertile population. So to this audience, I certainly don't have to introduce the problem of aging of oocytes. What we do know is that as females approach their end of their reproductive lifespan, the quality of their oocytes dramatically decline. And this is probably not due to one of the factors. It's probably due to multiple of reasons. We know that the chromosomal aneuploidy rates are skyrocketing. There is an evidence, and I'm sure Katalog will talk shortly about reasons why there is maybe a DNA repair problems in the aging oocytes. Dr. Keefe contributed to work on telomere shortening, and what my lab is really interested in is how mitochondria affect oocyte dysfunction. And, but all of these are not existing in isolating scenarios. There is always a crosstalk between these pathways. Now, to this audience, I don't have to say that the most fantastic cell in a human body is an oocyte. It's really a very unique cell that contains numerous maternal products that have been accumulated during the oogenesis in the form of proteins, RNAs, and organelles. Now, about 50 years ago, a group of developmental biologists started looking for genes that affect early embryo development, and they identified a group of so-called maternal lethal effect genes. And what these are basically maternally deposited RNAs and proteins that are usually not required for the oocyte formation and growth. However, they are required for the transition going from the oocyte into an embryo. Now, disruption of these genes really causes severe early embryo defects, and really the phenotype of the embryo reflects the genotype of the mother. So in these scenarios, the embryos themselves are heterozygote for any given mutation. However, it's the oocyte that is lacking certain compounds that then contribute to the phenotype of the embryo. And the group of proteins that had been identified so far that give these phenotypes, there is at least 50 by now, and they're all involved in protein degradation pathways, chromatin remodeling. There is also a group of transcription factors that are crucial for these pathways, and a group of really unknown proteins. So there is a great place for us as biologists and reproductive biologists and developmental biologists to go and mine the existing data to find out if any of these could be important for the human. Because to this day, we have not identified any mutations in a human that could contribute to what we call persistently early embryo phenotype, where the embryos stop developing in culture and just die. And no matter what we do, beside the ooplasmic transfers, we cannot really move these embryos forward. So the one great thing to work in Toronto is that there is tons of cancer biologists around, and they always go around and knock out genes. And then I'm really the only lab in Toronto that does infertility. So I get these phone calls from various labs saying, I have a knockout, it doesn't breed, please help me. So one of the collaborations that really blossomed from this kind of work is from lab of Tak Mak, who is a very famous immunologist and cancer biologist, who came to me and said, we have this mutant for a gene called TAP73, and we don't know why, but the mice don't breed. So we started looking at the TAP73, so what it is. It's a transcription factor that is involved in regulation of proapoptotic genes. It also has been implicated in autophagy, which is a survival mechanism. It also has been shown to interfere with differentiation of the cells as well as with cell cycle progression. Now, it's very complicated genes. There are two different promoters, and there are numerous splicing isoforms created. But for the purpose of my talk, I will be talking about TAP73, which uses the first promoter, the P1, and creates a transcriptionally active form of this protein. Now, interestingly, one of the most dramatic results that we have ever done and looked at is the fact that when we looked at young and old oocytes by immunocytochemistry as well by PCR, there is dramatic downregulation of P73 in the aging oocytes. And this is true not just for the mouse, because it has been already confirmed through microarray studies as well as through the targeted analysis that in the human oocytes, P73 isoform is downregulated. And this is not due to the silencing by methylation. So we don't know why transcriptionally as well as at the protein level P73 is decreased. So I already told you that these mice are infertile, but the question is, why are they infertile? And what we have, when we started looking at them, it became obvious that there are numerous reasons probably. Already at the age of the onset of puberty, these mice have a decreased ovarian reserve. There is about a third of primordial follicles that could be found in their ovaries. But most dramatically, they ovulate very few oocytes. And when you look at the quality of their oocytes, either if they are matured in vitro or in vivo, they have a high number of chromosomal scattering and really chromosomes misaligned all over the place. If we do an IVF, because normally these oocytes do not really fertilize in vivo, because the mice don't like to mate due to the problems that they have in the brain as well, we have actually found that the embryos are very poor quality. They tend to arrest anywhere between 4 to 8 cell stage, and only about 30% make it to the blastocyst stage. And the ones that do make, they have a very few cells and very poor quality inner cell mass cells. So why is this? What was really intriguing for us is the fact that there is this chromosomal scattering and abnormal spindle attachment. So we were very much interested in a process called spindle assembly checkpoint. And what it is, it's a mechanism that exists in somatic cells as well as in germ cells that ensures that the chromosomes are properly attached through the kinetochores into their microtubules. And this is facilitated by the existence of a complex of proteins that are called CDC20 and APC. So if all the kinetochores are properly attached, the CDC20 allows for APC to be degraded, and APC then itself is a ubiquitinase that otherwise would degrade other targets, and this allows the cell to proceed. However, if the microtubules are not attached, so there is a misalignment in a way because chromosome is not properly tethered, the CDC20 starts recruiting so-called SAC proteins, and there is a clump of about 20 different proteins, including proteins such as BAB1 and BABR1, which then do not allow CDC20 to interact with APC, and this means that APC will not degrade other proteins and halts the cell in mitosis or meiosis, and gives it a chance to either repair this attachment or commits the cell to death. So what we actually found is that P73 colocalizes with kinetochores. And if we then started looking through what happens with the spindle assembly proteins, what we found that while in the wild types, if the chromosome is misaligned, BABR1 complex does get localized to the chromosomes. In the mutants, BABR1 does not colocalize on the chromosomes, and you can see that the chromosomes are completely scattered and all the BABR1 is in the cytoplasm. Now, we have done some biochemical experiments and cell lines to show that actually P73 and BABR1 interact together. So to kind of summarize this section, what I have told you already about SAC, that it should normally form and arrest the cells and not let them proceed through the meiosis or mitosis if the spindles are not properly attached. However, if you don't have P73, the complex doesn't form properly, and the cell instead is allowed to proceed through the mitosis and instead goes on and forms these abnormal oocytes with aneuploid number of chromosomes, and these give rise to abnormal embryos. Okay, so while we were doing these studies with P73, and these are all published and have been done almost 10 years ago, Jury and Dean was way too busy looking for other maternal gene proteins in the mouse oocytes, and he identified so-called subcortical maternal complex. And what this complex is it's a group of cluster of proteins that are unique to the oocytes. They are only pretty much expressed in the oocytes. He always sees them together in a cortex of the developing oocyte, and he, by now, have knocked out every single one of these, and the phenotype is very similar to each other, which means that the embryos stop developing around 2-cell stage, they arrest, the mice are sterile. This is just one example from his publication of the TLE6 knockouts, and what you see there is that if you don't have TLE6, the whole complex falls apart. All the other proteins are reduced as well, which means that these proteins are required for the stability of each other. So if you don't have one, the whole thing is either heavily reduced or absent, and the proteins don't properly localize to the cortical region. Now, the rest of my talk will be about NALP5. Now, we got these mice originally from NIH, from Jury and Dean, as well as from Dr. Nelson. And the reason why I was intrigued by these mice is because this is the first animal model where you can see an embryo fragmentation after two-cell stage in mice. In a human, it happens all the time, but in a mice, it very rarely happens. And so I was really excited when I saw this, because this, I thought, will allow us to study what pathways regulate embryo fragmentation in the mice. And so Jury and Dean knocked out NALP5, and originally NALP5 was identified, or is a part of the proteins called NALP proteins involved in innate immunity. They are, originally it was found in a screen for autoimmune oocytes, and the oocytes are unique in that way that they mostly express this protein. Now, the mutants are sterile, I already told you that, because there is this two-cell phenotype. The protein itself is a quite large protein, it's 125 kilodaltons, and it has numerous domains that are involved in its function. So when we got these mice, what we really wanted to know is mitochondria metabolism, what happens to these mice. So what we found actually is that the NALP5 deficient oocytes have higher number of active mitochondria, but they're mislocalizing. They're not properly localizing to the cortex. And they have, overall though, fewer mitochondria, so they have more active, but fewer overall. And their redox state is altered, which means they have a lower level of oxidized FAD. Usually, if you have a mitochondria that are functioning too well or too much, that's associated with high level of reactive oxygen species, and this is exactly what happens in this model. The reactive oxygen species in the mutants are elevated, and this is also evident by marker by the two-cell stage of a protein called P66SHC, which is a sensor of ROS in the mitochondria. So very naively, we thought, okay, can we deal with the ROS in this model? And if we deal with the reactive oxygen species, can we push the embryo development? So in order to do that, we used advantage, or we took advantage of a small peptide that we supplement in a culture dish called BH4 domain of the Bclx protein, and we have previously worked with this small peptide and showed that it can improve embryo development as well as suppress reactive oxygen species in a normal embryos, wild-type embryos cultured in poor quality media. However, when we used this peptide on a NALP5 knockouts, it had absolutely no effect. It did not rescue them. We also started looking, could we use some other proteins that are peptides that could inhibit cell death? Would that help the embryos? And it absolutely did nothing. So that left us thinking that maybe it's not just a mitochondrial pathway, that there must be something else why these embryos are dying. So we went back and started looking, does really NALP5 only localize to a subcortical region, and can we find it somewhere else? And depending on a fixative, so this is a lesson from basic biologists, don't believe everything you see, and I love Jury and Dean, he does wonderful work, but depending on a fixative that you use, you can find slightly different results. So in this case, when we use a fixative that allows us to bleach out all the proteins that are not tethered to structures, we have actually confirmed that we can find NALP5 sitting on a chromosomes in a very similar place as P73 before. We also wanted to know what happens with NALP5 if we destabilize the spindle. So if we use nocardazole, which is a drug that will disassemble the spindle, you see that NALP5 disappears from the chromosomes, and it's not there in the knockouts in the first place, confirming that the antibody is specific. So then we started looking a little bit further, where can we find it? And it colocalizes with dynactin, and this is a marker of kinetochore. So very similarly to P73, NALP5 seems to localize to structures on kinetochores. So if it localizes to kinetochores, does it have any phenotype that relates to spindle problems? And so normal nice spindle would be shown in a barrel-like structures with chromosomes aligned, and voila, in a lot of the mutants we find that there is absolutely messed up spindle, bipolar spindle, scattered chromosomes, up to in about 30% of all oocytes. Now, this is similar to chromosomal scattering as well. Normally, I call these M2 oocytes as brush-like structures, toothbrush-like structures. However, in the mutants, the chromosomes are scattered all over the place. So I already introduced you the Sac spindle checkpoint pathway before, so I won't go through that again. So what do you see in NALP5 mutants is exactly the same problem as we see in P73s, where BAB1 and BABR1, the two proteins of the spindle checkpoint, are not localizing properly to the chromosomes. So if they're not localizing, they should have some biological consequences. And what are the consequences? We also looked at the P73 itself because I draw this analogy between the two, and we found that transcriptionally there is decreased amount of P73 as well as the protein in NALP5 mutants, and in a very small fraction of the oocytes, P73 does localize to a spindle. In most of them it does not, and it's all scattered. So, as I said, if there is abnormal spindle, this should lead to aneuploidies. Do we see any problems with micronucleation or an abnormal number of pronuclei? And that's exactly the case. We find that about 30% of all the M2s that are fertilized have abnormal number of pronuclei, and they're always maternal in origin. That was confirmed by the staining with H3K9 methylation. And subsequently, this also translates into formation of two-cell embryos that also contain pronuclei. And to the best of my knowledge so far, this is the first animal model that exhibits micronucleation in the embryos at the two-cell stage. And also you see that the cleavage division is not symmetrical, it's completely asymmetrical in at least 60% of the two-cell embryos. Okay. So, 60% are abnormal. 40% should be normal. So what happens to those 40%? Why don't they go further? And in 2008, a beautiful work from Japan actually showed that early pre-implantation embryos require to undergo process of autophagy. So sometimes between the zygote and 8-cell stage, a lot of the maternal proteins have to be degraded in order to proceed through the development. And if you mess up this pathway by genetically deleting genes that stop autophagy, you basically end up getting an abnormal embryo development and arrest anywhere between 2 to 8-cell stage. And one of the markers that's used to monitor autophagy is a protein called LC3. Normally it forms the foci in the wild-type embryos. However, in the mutants that are not capable of undergoing autophagy, it remains nuclear. So we then stained the wild-type and knockout NALP5 mutants, and what you see is that LC3 is accumulating in the nuclei, suggesting that autophagy is not functional. This was confirmed also by lack of lysosomal activity, which are organelles required for execution of autophagy, and accumulation of protein called Bclx, which is an instigator of autophagy. Okay, we were also thinking what could be the targets of autophagy at this stage, and we found no evidence that mitochondria could be the target in the mutants, because we actually see fewer mitochondria. If there is no autophagy, there should be more mitochondria. We didn't see accumulation of P62, which is a cargo protein that carries proteins to autophagy. No accumulation of ubiquitin aggregates, which is also target of autophagy. So what else could it be? So what we found, actually, surprisingly, is that the lipid droplets are something that embryos utilize to make energy. And what the lipid droplets do is that they most likely carry or are fusing with lysosomes to be degraded into simple lipids, which are then used for generation of ATP by beta lipid oxidation in the mitochondria. And so what you see in the graph is in the normal embryos, the number of lipid droplets decrease as they go from zygote to two-cell embryos. And if you treat two-cell embryos with inhibitors of autophagy, you see that there's accumulation of lipid droplets. In mutants, we see dramatic upregulation of the lipid droplets. And if they are not being used for autophagy, it should mean that they also have a less energy. And that's exactly the case, because you see that there is low level of ATP produced in these. Multinucleation is something that happens in human embryos all the time. I don't see human embryos anymore. I used to as an embryologist, but I know that statistics are probably not changing. At least 30% of the embryos are multinucleated, and we are struggling with, is it normal? Should we transfer? Should we not? There is some evidence that there are papers that say they're fine, other papers that say they're not. But we don't know why they are created and what can we do with them. So what I would like to tell you is that I think NALP5 could be one of the reasons why they are multinucleated and abnormal. ALK5 mRNA decreases with aging in a human oocytes. This is the data from our cohort 23 tubal infertility patients originally. As they aged, we used their oocytes and we found that they have decreased level of ALK5. But most interestingly, I would like to call upon this audience to start working on genes that we could use for patients' use to tell them that there is a reason behind infertility in some cases. Right now, there is at least 90 different SNPs that have been found in ALK5. There is no phenotypic data collaborating whether these patients are normal or abnormal. With so many mutations in such a large protein, there has to be a genetic reason for infertility in this group. The first paper came out about ALK5 and mutations only a year ago, and actually so far it's only linked to a group of cluster of patients that have epigenetic modifications in their children, as well as there is some evidence that these mutations could affect reproductive wastage because these families have increased rate of abortions. Now, we started looking whether ALK5 could be regulating epigenetics, and this is very preliminary data, so I'm not voting right now if it's true or not, but from our preliminary data, what it seems like it's happening is that in the normal embryos, and this has been known for years, the DNA gets demethylated over time, and the paternal pronucleus is the first one that removes the 5-methylcytosine and converts it into 5-hydroxycytosine, which means that the methyl groups will be removed and remodeled. What we find in ALK5 mutant embryos is specifically paternal DNA seem to be demethylated faster. If it's demethylated faster, perhaps there could be some errors that are contributing to later on epigenetical modifications that maybe we also see in IVF clinics. And with that, I would like to thank everybody to help to do this work, my collaborators, my students that definitely contribute and keep me on my toes, as well as all the patients that are willing to donate their samples for our work. Thank you.